This part is more about existing part collections and why measurement is important for all this part issue. Okay, let's start with an overview. I would like to introduce you to some pre-existing part collection outside from iGEM, which you can get also as an iGEM team. I would like to introduce you to pre-existing iGEM part collections, so in the iGEM community. Then we will come to the importance of measurements and how to document your parts. Okay, so I know this is a very crowdy slide, but it should just you don't have to look into the details. It should just show that for most of the chassis you are using as an iGEM team, they are super good published toolboxes out there. So for example, for yeast, there's this highly characterized toolkit with a lot of promoters and reporters and terminators. There's a similar kit for pick here. There's of course a kit for E. coli, and especially the plant community and algae community has adapted to that toolkit and a part way of cloning, and you can find all these collections on iGEM, which is a website where you can, or not a website, but a database of plasmids where you can order these toolkits for amount of money which is not so big for an iGEM team. So it's possible, I would say, for many iGEM teams, if you see something super interesting for your project there, you can order these toolkits from iGEM. So there's, as I said, there's for many bacteria, E. coli, Pseudomonas, yeast, algae, plants. There are toolkits out there outside from the iGEM community. Okay, and of course there are very, very useful collections, part collections, also in the iGEM community. And even very, very early on examples show these part collections. So for example, there's this Anderson promoter collection. And this is a collection for bacterial promoters with different expression strengths, which you can see here on the right indicated. And one is the kind of the relative standard, and all the numbers below are related to that one. And you have that for promoters, the Anderson promoters, then there's the RBS collection, the Anderson RBS, and also another RBS collection. There's a collection of already existing reporters on iGEM, and usually you could find them, of course, in the distribution kits. And I know that most of you don't have access to these distribution kits. So there are two ways of solving that problem now. So if you have an iGEM team in your neighborhood, kind of, which already participated in iGEM, then I'm quite sure that they are able to send you your parts which you are needed, because in the past they have participated in an iGEM, and these plates are usually laying around in some freezers in the university. So if you have an iGEM team in your country which already has participated in the past, perhaps just ask them if you can have some of the parts. That's solution one, which you can do to solve that problem, if you are able to work in the lab, actually. Solution two is you have a lot of DNA synthesis for free, because iGEM has this agreement with IDT, for example, and there you are able to just type in your DNA sequence and they will send it to your lab. And therefore, if you have an interest of some parts and you would like to use them for your project, then you can also order these parts if you're not able to get these parts from these collections. So these are the like two obvious solutions to the problem that you didn't get in distribution kit this year. So just ask around and use the DNA synthesis, I would say. Perhaps the other iGEM community committee member, have some other ideas, but let's come to that in the question section. Okay, now I would just highlight some iGEM collections or projects which created some part collections, but this of course doesn't mean that there are not a lot of other super good characterized part collections which I forgot. This was just kind of obvious examples because they won, for example, the part collection prize. So as I said, I don't want to say that any other project is bad or something like that. Just a few examples which could help you for your projects. And the first example I would like to introduce you is from 2017 from Vilnius, and they had the project of Sinori, and they created a part collection of origins of replications in bacteria, so that you are able, for example, to control the copy number, that you are able to split an origin, that you are able to split antibiotic resistance cassettes. And if you're interested in that, in copy number and stuff like that, you should look into that project perhaps as one of the examples. Another project which I was also involved on, so if you have questions on that, you can also ask me or write me an email. You can find my email on the iGEM website. Is the Marburg collection from 2018. Actually, we did work on Vibrio nitrogens, which is the fastest growing organism in the world. But these parts were originally designed for E. coli. So we didn't create a specific toolbox for Vibrio actually, because we just took the E. coli parts of it. And this is a kind of comprehensive collection of different parts which you can use for your projects in E. coli or any other bacteria. These promoters in RBS and so on work in a lot of different chassis. And if you are interested in that, I would also explain that more in the next week in the Moklo section. So this is just another example, 2018 Marburg part collection, if you're interested in that. This is more like very, very broad and general parts, not very specific parts. Okay, now what I would like to come to, why is measurement important for these parts? Why do we need proper and standardized measurements for these parts? And the reason for that is, if you have these parts, for example, in this case, on the picture, you can see yeast promoters. It doesn't matter what it is, not right now, but if you have these parts defined and characterized, they are much more valuable for future iGEM projects. Even if you have worked with, for example, I don't know, PET degradation or something like that, if you have kind of values for these promoters or ribosome binding sites or whatever, other teams could use these characterization and their characteristic in different conditions for their future projects. And that's quite important. What I would like to show here, that is really important, not just for your project, but for the iGEM teams and the community, that you have proper measurements which facilitate more complex genetic designs for other teams. And examples, of course, for that would be promoters, RBS, terminators, and collections for all these things in different chassis or something like that. And that's very, very important, what you should keep in mind. that's more about other teams and for the future of, or if you want to have other teams your part that they use your parts, then you should have proper measurements. I know that that's not the topic of that webinar actually, and Jake will also explain that or in his section, but I would like to mention it anyway. So there are, for example, different ways of showing your results, right? And showing your fluorescence. And you should definitely look onto our measurement page, how to measure stuff, how to measure fluorescence, how to measure OD and things like that. Because usually when you do your plate reader measurements, for example, you get relative fluorescent units. And if other teams would like to use your parts, which you used in your project and have kind of characterized, they can't compare these measurements when you have these relative fluorescent units, because these units doesn't say anything. So if you put these samples in a different plate reader, you would totally get different results than in any other lab. So it doesn't make sense to show these relative fluorescent units. And just to mention it here, but as I said, Jake will explain that in a different webinar. You should use absolute units for your fluorescent measurements, because then your parts which you submit to iGEM have much more value, because teams can compare these results to their results and also make conclusions out of that, because otherwise that wouldn't be possible if you have the relative units. For further information, visit our measurement page and look into that. That's a really important topic in iGEM actually, that teams shouldn't use relative fluorescent units, and make your graphs and your results comparable to other teams, because then your parts have much more value. Another important topic, and that's also something to my heart kind of, is part documentation. I will show you that in a minute, how this works, but you have these registry pages. So the registry is the collection of all the parts, and that's the web page where teams would look up parts for their future projects. And if you want parts to be valuable and to be usable, then you should, on the one hand, have a proper description that you can understand it easily what these parts do. You should have in general, you should have your parts sequenced, because if your data show something with a part which is not sequenced, and perhaps it has a little bit different sequence, then your data actually are not valuable, or you can't use these data for a part which has a different sequence. That's why I would say every part what you created and you use for your data has to be sequenced. And in the end, you should have all your data for a specific part, let's say a promoter, a coding region, whatever, should be on the registry page. And that's a mistake what many iGEM teams do or did in the past actually, because that's also mandatory for or was at least mandatory in the past for the medals. And if you have all your data and your plots just on the wiki, this is not enough. And the reason for that is, if teams are looking for parts they would like to use for their project, they wouldn't scroll and look into all the different wikis which exist. They want to have all the information on one web page. And that's why it is important to upload your data and your description and your part sequence and so on the registry page. And you should do that also for all of your parts which you used in your project, and not just for one or two and the rest is not so important and so on. You really should spend time on that. And also one advice from my side, I know it because I was also an iGEM member in the past, you shouldn't do that in the last minute, because this takes so much time in the end, and then there's a deadline when the registry is closed, and in the end, yeah, you are not able to enter any information into these registry pages. Okay, the last part is just that I would like to show you how you can find the registry parts, and a little bit about the registry I would like to show if you are totally new to iGEM, because when I started iGEM, I was not so familiar with that. Yeah, oh, I closed it here, but let's start it again. So you just type in iGEM registry, and I'm sure that you can find it. And then, okay, this is web page here above here, and I know that it looks a little bit different than the other iGEM pages, but you have, for example, the catalog here with well-documented parts. That's also a nice collection for your project, and you can find, for example, promoters and RBS and coding region terminators and so on in these on this web page. And I would like to also show an example page. Let's see. It's not. Oh, it's opening. This is one item, one registry page, and you can find some information here. You can find data here. It doesn't look very nice in some BioBricks, and in the end, you also have to fill in the information for the sequence. If you are looking for parts in the registry, there's an information box here, how many times it's used and where you can get this part. And that's something many teams overlook perhaps. So when you click here on get this part, you can see the exact position, the exact well, and the backbone, and if it's sequencing confirmed for that specific part. So if you, for example, have the problem in this year that you don't have access to these distribution kits, then you have to write down this position and this plate, and for example, email some teams if they have the 2017 or 2018 distribution kit plate, and if they can send you the part from the well 17P or something like that from kit plate 4. So this is the way of working with the parts in iGEM. Yeah, so this was just if you want to get the sequence for your part which you are interested in. Unfortunately, there's not a download link for plasmid maps, but you can get the part sequence by clicking on here, and then you have just the part sequence, not the plasmid itself. So you would have to insert that into a plasmid map for BioBricks to get the whole plasmid map. That there you can get the sequence information, and that's also the web page where you have to enter your parts. So if you would like to add new parts, of course you have to be logged in. So I will do that quickly. Let's see. So here you have to create a account, but that's easy. I don't have to show that actually, I guess. And then you can edit every existing page, but you can also add new parts to the registry. And that's something you can find here in tools, add a part, and then you have it guides you through the process of adding a part. If you're a basic part is something what I mentioned, so with the defined function, so for example promoters, RBS, terminators, some one small part. A composite part is kind of a piece of DNA or plasmid which you assembled from these basic parts. So it's kind of an assembled piece of DNA or a composite part which is comprised of, for example, promoter, RBS, binding site, protein coding regions, but this can vary. So it doesn't have to be a full transcription unit. A composite part could, for example, also be promoter, RBS, or something like that. And you have to differentiate between these here if you want to add a new part. And then you can just click on that here. Then you have to enter which team it should belong to. And here I have a lot of different teams because I was member in all of these. But you would probably just have one. And then you would enter all your description and all the information for your parts in here. And that's something which you have to do for all of your parts. Yeah, and I guess the rest is similar to the wiki. That's something I wouldn't show here. But for me, it was important that you understand the registry a bit and that you know how to enter a new part for your project. Okay, let's come back to the questions. And yeah, let's see. You can ask now questions.